I'd like to thank the Academy of Sciences for inviting me to give this talk, and I'm happy to be here. Is this a pointer?
Oh, yeah.
The screen one?
Yeah.
Top, okay. Yeah, I'd like to thank my fellow authors in this publication. This one.
Maybe
Yeah.
you want to be
Oh,
closer to
no,
the
that's-
microphone.
How do I go back?
So this is the laser.
Oh, that's
That's it. Towards that. Okay.
Huh?
Towards the back of the room. So the slide
Oh,
forwarding.
okay.
Yeah.
And this is back, right? How do I go back to the first slide?
Can you go back to the
First slide?
Yes.
Should have practiced this before I got up here. Sorry. Okay, I'd like to thank Jin Liu, who worked fairly closely with me on this project, and Arnold Tesoro. And I'd also like to thank the sanitation districts for funding and supporting this effort. So just to give you some background on how we got involved in this project, I know that we are a POTW, and we are supposed to be one of the bad guys, right? The plants were supposed to be discharging lots of microplastics into the environment, and everybody just thought that it was all coming from the utilities. I think today I will try to make a credible case that we are actually one of the places that can limit and minimize microplastic to the environment. So that would be my goal here today. Hopefully you don't start laughing, and I can convince you of that fact before this talk is over. So essentially, this was discussed in some detail yesterday. There are two basic types of plastics. There are thermoplastics and thermosets, and the differences are clearly outlined here in the two columns. The thermoplastics are the ones that we have seen a lot. They can be recycled. The thermosets are not so likely to be recycled like tires. They are formed in situ, the final product, and you cannot easily recycle them. You can convert them to different products, but you cannot recover the original compositions. And the varieties of plastics, I think that has seen some discussions. The most commonly available commercial types of plastics, polyethylene, that has been talked about quite a bit, methacrylates, PET, polyvinyl chlorides, polycarbonates, polypropylene, and I think most of you here are very familiar with those. This is an example of a typical wastewater treatment plant. There are 3 stages. There's a primary stage, secondary stage, and in some cases, tertiary treatment that takes place. And you guys are wondering how we got involved in studying microplastics, and I think it was, just to give you some insight, about 5 years ago, a group of reporters from CBS descended on our facilities and started asking questions. And we had never considered looking at microplastics. I mean, it was not something on our calendar at all. I mean, we had lots of stuff on our plates, CECs, we had, PPCPs, just you name it, compounds of emerging concerns in general. We were looking to reduce the reporting levels, and it was not something that we were ever considering. When the reporters came to the facility, they started asking questions, and we had no answers. And the way things work at a utility, any type of pending regulation creates massive amounts of concern because it's costly. So the engineers started calling down to the lab. I mean, we had no answers. We had never looked at particles. We had no idea of how much was getting into the plant, how much was leaving the plants. So it became somewhat of a hysteria. Engineers started coming down to the lab. Do you guys have methods to do this? So we started looking, and there was nothing available. So there was a crash course or crash program that we undertook at the time, is to find a way to determine how to characterize these plastics coming into the lab. My background as a synthetic organic chemistry drove me in one direction. Well, maybe I can get something, spike it into the plants, and then we can study it intensively for a week, and I'll have all the answers. But I didn't get very far with that proposal. They wanted nothing spiked into the plant because at some point somebody might discover that we put microplastics into the plant, and that would create a lot of problems later for us. But I persisted and went ahead and ordered some of these microplastic particles. We found a company that was in Santa Barbara, which was not too far away from where we are. And I thought, gee, this is fairly simple. I'll just spike, if we could have, I would just spike this in the front end in the wells, and then look at how it moves through the facility, and then we'll know exactly. We had different sizes. It was fluorescent. It would be fairly easy to determine if it moved through and what sizes were trapped and what sizes were retained. But we didn't get very far with this. Well, we still thought that this was a good start. I mean, and yesterday somebody mentioned that spheres are not the best way to actually do a plant study or to do any type of study. I think there is some value to using spheres. They have the smallest surface areas, and if they're going to be transported or moved through a plant, these will be most likely to make it through to the final. So we started looking at how these particles would behave in this system. So initially we spiked them into the water and looked at the distributions. About 60% of the microspheres floated. Some ended up on the bottom. As you can see here, these made it to the bottom, but the majority stayed on the surface. So that was the first step. How do these microparticles actually behave when they're in solution? So the next thing we did, we looked at the mixed liquor and shook them vigorously for about probably 5 minutes just to be sure we had them completely mixed, and then sat them on the bench top and observed what happened. And we saw the same sort of behavior. About 40% floated to the top. The rest got stuck in the flock. This was after 10 minutes. And then following this, after an hour, you can see that we had the partitioning where most of the I guess about 40% of the microspheres stayed on the top. Some was trapped in the, some got trapped in the actual flock itself as it settled. We also wanted to see what impact things like surfactants and solvents will have on the distribution of these particles. So this was one of the bench tests that we did. You can see again in water, you had the primary distribution on the top. If we added it to, say, T-butanol, we can get a fairly uniform distribution in the sample itself. And then you can see the impact of surfactants and surface wetting will again have the distribution more uniform in the water on the aqueous layer. The one approach we thought we can apply in our study was to do a filtration to segregate different sizes of particles. So we wanted to be sure that if we use a sieve, we can actually isolate the particles effectively, where we had a few surprises around the corner. So this showed that the particles did not get through a mesh size that was smaller than the particle size. So this sort of worked as we expected. When we mixed the microparticles, what we saw was a little surprising. We could not use this technique to segregate and isolate different fractions of sizes in the plants, because what we see here is that the particles clump together. Again, they are hydrophobic, so they are more attracted to themselves as they are to the aqueous matrix. And so it's possible then that if we use a mesh size that's larger, we can still be isolating and collecting particles that are of much smaller dimensions. And this is what happened when you have these mixed particles in water. You can see that they don't uniformly spread, they clump together, and this is exactly why we couldn't filter them and isolate them. This one looks like a photo from the Hubble telescope. You can see again that they are all sort of clumping together. And so this told us one thing, that we could not use techniques, just simple filtration, again, to isolate and segregate particles. And again, as part of our study, we discovered that the toothpaste micro-polyethylene particles were very abundant in our wastewater effluents because at the time, I think it has since been banned. This was applied to toothpaste. I guess it made it a bit more attractive to people buying it. It looked like it had like some special whitening ingredients, but it was just colored polyethylene. We actually contacted somebody that worked for the company that told us, oh yeah, we just ground up polyethylene particles, dumped it in the toothpaste, and it looked great. You paid an extra 50, maybe $1 more for it. The other thing that was noticeable is that these toothpaste particles, if you look fairly closely at it, you'll see there's some hints of blue in here. They were very heavily there was micro depositions on it, biofilm and growth, and this was very persistent in a lot of the samples that we found. So again, the first step, if you're going to be doing these types of analysis and isolations, you have to find methods and means of removing these biofilms that accumulate in the samples. One thing that we should point out, we have two sets of plants, and we break them into two groups. We have upstream plants that do not handle solids, and then we have a major plant that's downstream, which is our largest facility. All of the solids from the upstream plants are taken, removed in the early stages, and sent down to the joint plant in Carson, which is about probably 15, 20 miles away. And that's very relevant in terms of what we observe in these samples. It takes a period of time before these biofilms can grow. In the upstream plant, the SRT is much shorter. So this type of growth was not evident in the plants that were upstream. They just didn't have the time, the time of transmission and the time that it's spent at the joint plant where the solids are handled allowed these growths to take place. And this had a huge impact in terms of the number of particles that we were finding at the different facilities. Again, here you can see the early stages of biofilm. It's fairly clear, and you can see the different colors that we isolated. There were techniques that we used, such as the Fenton reaction, to remove these. They're very strong oxidizing conditions. The other thing that we observe is that the surface fouling was very prevalent in all of the plants upstream. This is the first point where the samples, where the actual effluent, the primary effluent enters the plant. These particles are closely associated with any of the grease, fats, and oils that are coming into the plant. They are all hydrophobic. It makes no difference what size these particles are. They behave exactly the same in terms of the plant processes. So if the particle is 1 nanometer, it gets into that layer. The densities is what the functional separation means is. So the waste comes into the plant, floats to the top. There are flights of skimmers that come by and scrapes the surface of the water. That gets removed from the treatment stream and sent down to the joint plant. So there is a misconception that size has something to do with the ability of plants to separate microplastics. And I can say to you categorically that that's not the case. It's treated the same way as if you have a very small droplet of oil. We have lots of fat from domestic waste and restaurants that come through the plants, and they run the range from very small, minute droplets to big clumps of it. And it's all in that one group that we see this material that's associated with the microplastics. The plants do not see or do not differentiate in sizes, I must emphasize. We also looked at fiber residues. I think one of the criticisms that we had is that we did not pay much attention to fibers in our process when we analyzed the final effluent concentrates and the particles that we were collecting. I think there were comments that we didn't spend enough time, or we didn't have the ability to look at these microfibers that were coming through. We did not see any microfibers in our analysis. What came through the plants, fully digested, we used the Fenton reactions. We eliminated all of the background. The materials that we collected in our microsieves at the very end of the plant were very heavily laden with residual microbes from the processes in the plant. I mean, it's not like we did not have the ability to see it. We would essentially, when we filtered, let's say, 100,000 gallons, we would have enough residues in those that exceeded, maybe 500 mils, and then we'll have to treat those residues. And we were treating lots. There were lots of rotifers. They were the most abundant. So it's not that our techniques were deficient. We could see it if it was there. When we take this through a full oxidative digestion, we had clear solutions at the end and some minor residues. And we could look at those and extensively examine them, just like we did with the microplastics. We did not see any occurrence or incidence of discharge of those. And again, one of the criticisms that we have is that maybe we are advocates for the plants, or I was an advocate for the plant. That's not really true. I mean, when this dictate came down from up the hill, as we say, and they said, yeah, you guys got to do this assessment and do it thoroughly. We have to have answers. As chemists, the only benefit that we have when that sort of thing happened, again, there's a great fear that if there's a pending regulations that it would impact us. And so all the engineers, the ones that you never saw for months that slept at their desk, suddenly comes down to the lab, what are you guys going to do about this? And that's the time we get even. Then we can say, well, I need a triple quad, I need a time of flight if you want to see those. So that's how we play on this. We basically use that as an opportunity to get some great instrumentation. So that's how we get even. So we applied all of the techniques that were necessary to determine what was in our discharges. We were very dedicated and very involved in getting the right answers. It's not like we were advocates. We wanted to know what was really in there. I think it served our purposes that at least this is something else that we discovered, we wanted to know more about. And so if you look here, you can see, I guess you guys possibly can read. We looked at fibers from like washing machines and lint traps and all of that, and we compared them. We know if they were there, we should have been able to see them and see them readily because they have different appearances. We did apply techniques, just like everyone else that's doing studies now. I know we were a little ahead of the game in some cases, but we applied all of the techniques that we thought were possible at the time in order to evaluate this. This is a surface skimmer that we came up with. It's an idea because at first we thought we were getting samples that were taken from below the surface. The intake pumps were collecting samples from the wares that were subsurface. And since we observed that a lot of these microparticles were floating, a lot of them were, we did not want to miss that in terms of our sampling efficiencies and our sampling representativeness. So we had this design, and it worked fairly well. I mean, if there were particles in the surface, we obviously would trap them at that point. And we could also assess the volumes that we were analyzing just based on a simple calculation. So this sort of worked out well for us in getting a more complete picture of what was going on at the plants. So now we come to that $64,000 question. Where are the particles going? And, this is a breakdown of what we found. Again, as I stated earlier, the primary tank skimmings showed the highest concentrations, the highest counts. We were trying to put some units to this, but it was not possible to do that because the way the skimmers work, we did not have a direct relationship between the amounts of solids that we collect at the influence side and the volumes of sample that represented. So we had to have other indirect ways to determine what the concentration of particles coming into the plants were. We had scum in the aeration tank, and there was a similar issue here because in the aeration tanks, the stuff that rises to the surface could be resident in that tank for a week, a month. We didn't know. It was not something that flowed along with the inflow and flow. So we had no way of putting a unit to this concentration. The return activated sludge were a big surprise for us because we did see the particles were being proportionated into the settled flock phase. So we assumed that at least maybe 20–30% of the particles would be resident in the return activated sludge, and we would pick up a fair amount of that in the residue. But those numbers surprisingly came back very low. We had 1 particle in 20 mils of in the RAS. And again, that was one of the surprises that we had. The secondary effluent, and we did this at 7 different wastewater treatment plants, upstream plants. The secondary effluent, we found 1 microplastic particle in 15,000 gallons. So I'm saying that this process is a pretty efficient one, just based on these numbers, and there is not any bias here. We were just very surprised. When I wrote the manuscript for this, I used surprise like 4 times, and one of my bosses edited it, and he said, surprise, it's not so scientific. Let's get some of that surprise out of here. I kept something in the conclusion. So again, the secondary effluent was surprising to us because we assumed all along that there was some transmission all the way through to the filters, because I think that was the theory that was prevalent at the time, that the filters are not going to trap this stuff. The particles are too small. But I think there was an awakening here that the filters actually had no role or no appreciable role as far as we observed in the plant, because the next one that came up was the backwash. And anybody that's familiar with the plant, daily the filters become clogged with residues, microbial residues, and they settle, and so there's some sort of buildup of pressure on the filters. They have sensors and stuff to determine when the filters need backwash, and it's typically done daily or every 2 days, depending on the plant. And we had some experience with this when we looked at NDMA fairly intensively at the plant, because we were looking at what was converting some of these precursors in the plants to NDMA, which was a big issue for us. The highest concentration we found was actually the residues on the filter beds, where the polymers that ended up residing from the flocculants, and that's where the highest formation potentials were. So I would assume at that time that if we're going to find any particles, we should look at the backwash. So initially, when I sent my chemists out, we took samples, and it came back, and there was no particles. And I said, no, you guys must not have sampled this correctly. Let's go back and do it a second time, a third time, and a fourth time. And sure enough, there was no micro particles there, no microplastics. So I think this in itself was a discovery, because all along, again, as I said, the assumption was the filters were the point where the plastic residues or microplastic particles were being removed. And that was not an efficient process, and that was completely wrong. And then in the final effluent, we filtered large volumes. This is the thing that we did first before anybody else attempted. Most people were sampling, they were taking 2 liters, 4 liters. I wanted something that was statistically representative of the true flows in the plant. So we really went all out. We sampled sometimes for days on where we tapped into the flows. And we were not finding any microplastic residues. I mean, very little, if any. And some of that could have been incidental contamination just from air and other types of transport means. And again, in identifying these, we use systematically pragmatic approaches. I mean, I know that now there's a sort of a trend to go to more and more sophisticated type of instrumentation, FTIR, Raman. I think we did a fairly adequate job by applying these, and I would recommend then that as we progress towards coming up with techniques for doing and identifying microplastics, that we should not throw these away. This came in very handy. If you guys ever worked or looked at treatment plant samples, they are very, very complex matrices, and it would just have taken us maybe 10 times as long just to do a full assessment if we were doing it instrumentally. Just to give you an example, at the influence side, you get so much paper residue coming in from toilet paper that it just overwhelmed any technique that we applied to doing matrix removal. We essentially threw our hands up. We started with a 1 millimeter sieve, which was a very large to take out the largest solids, and then we cascaded down in the tray. It didn't last even 15 seconds before it was completely clogged. So it's not practical to apply any of these techniques. And we, as a hazardous waste certified laboratory, we handle matrices that are very, very dirty and complex. There is nothing that comes close to an influence sample, trust me. You've just got, it's just very bad. So all of these, like the acidic digestions and stuff, it was overwhelmed. And there are big concerns now about microfibers, and I think maybe I should I think my time is up too. There is nothing that compares to the volumes and quantities of cellulosic fibers that are coming into plants. Any plant or any facility that can handle that level of solid fibers will have no problem handling washing machine discharges. We attempted to do, and I know my time is up, some quick bench studies where we mixed fibers from lint traps with the toilet paper. We generated like an artificial toilet paper solutions. They mix intimately, they do not separate. So the same mechanisms that are involved in isolating cellulosic residues are very applicable to isolating any type of laundry or polyester type residues. Okay, and with that, let me just go. Okay.
Time for maybe one quick question if someone has. Go ahead, Greg.
Actually, it's a two-part. One is, where do your solids go after it leaves the plant? And the other one is, how well do you think your agglomeration of the particles clogging up your sieves, how well do you think that applies to nature? Again, random sample out in the surface water.
Well, I think the lesson that we can take from, I think the lesson that you can take from that is that we have to focus on generating homogeneous distribution of plastic particles before we can do true analysis. It's like any type of organic analysis. You better be sure you have a representative sample, and that was one of the things that we had to address fairly early when we were getting large quantities of the toothpaste polyethylene samples. We had to have a way to manageably determine counts and stuff, and one of the approaches that we have was to add T-butanol so we can get an even distribution and then take a small subsample so we can do true counts. And your first question was?
Solids.
The solids. Yeah, that's something that we have to deal with, and the argument that I made as to how wastewater treatment plants do minimize or reduce the counts of plastic, about a half of that solid waste is taken to landfill. The other half that's land apply, we really don't have any answers in terms of how stable that matrix is, that mass is, and how long it would take that to get back into the environmental contamination cycle. But that's a study that probably my plant engineers do not want us to do.
Thank you so much.
Because anything we discover from that canon will be used against us. It's like a Miranda's right.
Thank you. Okay. Thank you very much.